This is Dr. Zalzala, Chief Medical Officer, AgelessRx. I'm proud to present today the Pearl trial webinar, how Pearl participants can help interpret their results. I know there have been a lot of questions about this, so we thought we'd shoot a webinar, or a series of webinars, I should say, in order to help our clinical trial participants explore and interpret their results. So just a brief intro on me for those who are not familiar. I'm Dr. Z, people call me, Chief Medical Officer of AgelessRx, also the co-founder. I am the clinical investigator for the Pearl trial and have extensive telemedicine background, which allowed us to take this trial and make it decentralized. For those who are not familiar with Pearl, it's a double-blind, placebo-controlled trial. There's 3 arms in it. 5 milligram rapamycin, a 10 milligram once-a-week rapamycin, and a placebo. We were shooting for about 150 participants, so about 50 in each group. And our primary endpoint was visceral fat by DEXA. However, we have several secondary endpoints that we'll talk about, including DNA methylation test, GlycanAge, PhenoAge, and various other safety outcomes as well. And we expect to wrap up the trial by the end of 2023, and shooting for publication maybe around middle of 2024. It takes time, unfortunately. Today I'm going to be talking about the kind of the 3 parts. Part 1 will talk about safety, the Pearl outcomes, and things like that. Second would be more like the efficacy of the Pearl trial, and the third is the what's going to go on after the trial is completed. So part of the safety, I'll be talking a little bit about the timeline, the endpoints, and the biomarkers we're using. Talk a little bit about the purpose of each test, why we selected the tests that we're testing, and also a little bit about the subjective data that we're collecting as well. So just to clarify on the timeline for the Pearl trial. At baseline, we asked all participants to do blood work through our partner Quest, a DEXA scan through one of our DEXA scan partners. This is a body composition scan. There are several at-home tests that we asked participants to do, such as GlycanAge and Thorne, which is a stool test. A subset of our participants underwent the TruDiagnostics methylation test. And then optional, we asked patients to optionally be involved in our biobanking, as well as filling out dental forms, and one of our partners called GeroSense. And so that was kind of baseline for everybody. At 2 and 4 weeks, we collected blood work, again from Quest, from our reference lab. This is for more for safety, or just to make sure that we weren't causing any abnormal blood markers, elevated kidney function, elevated liver function tests, things like that. And then at 6 months, we had most of the baselines repeated, and the 12 months as well, and including the surveys. So what did we ask for from our reference lab? A lot of the biomarkers we were looking for were standard biomarkers, kidney function, liver function, lipids, because, it's thought that rapamycin can disturb the lipids, at least temporarily, A1C, blood counts as well. Again, there was concern that maybe rapamycin might be affecting those. And then as part of the lipids, we're looking at ApoB as well, which is a little bit more of an advanced lipid marker. And like I said, we were looking at these biomarkers the most important thing, we were screening to make sure somebody didn't have some kind of undiagnosed anemia, undiagnosed kidney or liver problem, or undiagnosed diabetes, metabolic problem. And then B, the other one was more for once somebody kind of cleared those screenings, then we repeated them over time to track for safety. Like I said, we wanted to make sure, for example, rapamycin wasn't causing a huge elevation in cholesterol, or an elevation in blood sugar, or disturbance of subtypes in one of the blood counts. So that's kind of part of what we use to measure safety. And in terms of what these blood tests are, we broke it down to several categories. For metabolic function, we would use things like glucose, A1C, and then our DEXA scan gave us information on lean mass and visceral adipose tissue. Cardiovascular risk would be more the lipids, triglycerides, LDL, ApoB. Looking at the function of the bone marrow, or the circulatory, the blood in circulation, we'd look at white and red blood cells, as well as the hemoglobin inside the red blood cells. Organ-specific, like I mentioned, kidney and liver, we would look at creatinine, ALT. And then more experimental or more like longevity-focused biomarkers, such as GlycanAge, DNA methylation test, the GeroSense, and then the Biobank, I'll be talking a little bit more about that. And then, of course, quality of life surveys, such as the SF-36 and the WOMAC. I'll talk a little bit about those as well. And then other longevity or health-related outcomes, for example, the dental forms, the gut microbiome testing, and PhenoAge. Specifically, diving a little bit deeper in terms of some of these measurements, so triglycerides are a marker of metabolic health. They're a main constituent of natural fats and oils. There are a lot of things that can elevate triglycerides. Elevated triglyceride tends to be a signal of poor metabolic health, and vice versa is true. The lower your triglycerides, typically, the better somebody's metabolic health is. It was known prior to the study that rapamycin could increase triglycerides, but we think it's temporary. Might have something to do with the kind of the turnaround of autophagy and the turnaround of lipids and things like that. But right now that's kind of speculatory until we get more data on it. Total cholesterol is kind of the total count for LDL, HDL, and then various other cholesterol particles. LDL is low density lipoprotein. A lot of doctors call it the bad cholesterol because that's the cholesterol that's thought to line the arteries or get built up in the arteries as somebody gets older. It has to do with damaged LDL. LDL itself doesn't get, but damaged LDL. And the idea is the more you have in your circulation, the more likely it is to get damaged, the more likely it is to end up in your arteries and cause problems. It's not a direct measurement. Almost all reference labs, it's what's called a calculated LDL-C or calculated. Glucose is the main sugar found in blood, and a lot of people measure that over time, and that's that. We're using that as one of the measurements for metabolic health as well. Other markers we're looking at, creatinine. It's used as a measurement for kidney function. Your body makes a certain amount of creatinine per day, and your kidney is tasked to clear it. And so if we see a buildup of creatinine in the blood, that means the kidney's not clearing it properly, therefore it's not working properly. You don't want too low of a creatinine, but you don't want too high. You want to kind of right in the middle, like a lot of these markers. ALT is an enzyme that's made in the liver. A small amount kind of given off in the blood, just as a normal turnover of the liver cells and things like that. But if the liver is irritated or being damaged or somehow affected, usually one of the first signs of liver damage is an elevation in ALT, or AST is another marker. But usually they're redundant, either the ALT or the AST. So the A1C is thought to be a 3-month average of your blood sugar. Basically, it's a percentage of how much of your hemoglobin has a glucose molecule attached to it. And the higher your blood sugar is over time, the higher that non-enzymatic attachment of the glucose to hemoglobin. And so a healthy A1C is around 5 up to 5.5. Once you get above 5.7, 5.8, then that you start trending towards prediabetes. Once you get above 6.4, then that's considered to be diabetes, in which case your average blood sugar is running far too high. WBC, or white blood cell count, is basically just a measure of how many white blood cells were found in the blood, and it's a standardized measure. Variety of things can either elevate the WBCs or reduce them, and that's kind of why we look. It's thought to be one of the markers for immune function. On the flip side, the red blood cells, it carries, the red blood cells carries hemoglobin in it, which carries oxygen. So those two are kind of linked, and we look for those markers as well. And then ApoB it's thought to be a more accurate marker for cardiovascular disease. Basically, almost every what's called atherogenic particle, or every particle that's responsible for atherogenesis or causing heart disease, is thought to carry an ApoB on it. And so if you count the number of ApoB, or if you get a concentration of ApoB, it's thought to be better than LDL, because LDL you can have large fluffy LDL, you can have small dense LDL, and different size LDL particles can mean different things for people. So ApoB is kind of an additive marker in addition to LDL, and it's thought to be a more accurate, or especially when added to other markers like triglycerides and whatnot. So that's why we included in this trial, just to get a more accurate view. Because you can imagine a situation maybe where somebody's LDL goes up, or their ApoB maybe goes down, for example, and it could be at that point. If you just looked at the LDL at that point, you said, oh, this is bad. But if the ApoB went down, for example, then you can say, oh, that's actually good. Because the LDL looks like it's a higher concentration, but maybe doing something good. But again, that's just speculation. That's just an example. Here's an example of if you were to get your report from Quest. A lot of times you can go on the Quest portal and get it, or sometimes we'd offer it to the patients. But this is how the lipid panel would look with the total cholesterol, HDL, triglycerides, LDL, non-HDL. And various different ratios and references. You'll notice with the LDL, they give you different references kind of depending on your risk factors. So that's why there's a lot of text here for it. But I just wanted to familiarize patients with how just standard reference blood work is reported. This is what's called the comprehensive metabolic panel. Some people call it the Chem14. It's approximately about 14 markers that we look at, from glucose all the way down to electrolytes, protein, and liver function. And again, each one of these has reference ranges, and you don't want things to be too high, too low. Usually you want to be in an optimal range as much as possible. This gives you the reference range. There are other resources you could look at for optimal range. I'm not going to get into those right now. Because optimal ranges are kind of debatable. Reference ranges are set by the lab. Optimal ranges are set by more like expert opinion on what optimal should be. A1C we talked about before. This reference range is anything less than 5.7 is good. If you're between 5.7 and 6.4, then they consider that pre-diabetes. And if you're above 6.4, at or equal to 6.5, then that's kind of the cutoff for diabetes. Blood counts, again, you'll have various different blood counts in the subconstituents of, especially of the white blood cells, and the various different reference ranges. As long as you kind of fall within the reference range or close to it, then there should be nothing to worry about. Minor abnormalities come up all the time. This particular patient, for example, had a slightly elevated hemoglobin. Maybe he was dehydrated. I'm guessing it's a he, because these are just guys who have a higher hemoglobin. Women tend to run a little lower because they typically have their monthly cycle, or they have other reasons. Maybe they don't eat enough, as much red meat as men do, or don't hold on to as much iron. I'm not going to get into that discussion, but in this case, I'm just kind of showing that this person had a slightly elevated hemoglobin, 15.7. Not much to write home about. It's flagged as red and high, but you look at the numbers, it's not that much far off. Maybe in this case, I'll tell the patient to either cut back on iron if their iron levels are high, or go donate blood, or try to figure out why the hemoglobin is elevated. There's several reasons. ApoB, this is how it's reported. Again, just like LDL, it kind of gives you categories depending on what your risk factors are. PhenoAge. So, using the standard blood test, you can pull out 9 markers from the standard blood test, and there's this calculator developed by Morgan Levine at UCLA. Morgan Levine and a group at UCLA. Basically, what they did, they looked at biobank data from over 11,000 people, ages 20 to 84. I wish it was 100,000, might give us more accurate data. But basically, they kind of looked at what blood values, what markers in standard blood tests could they look at to help predict mortality and disease. And so they came up with this algorithm called the PhenoAge. And so when you calculate your PhenoAge, and AgelessRx has a free PhenoAge calculator, you just plug in your blood work. But when you calculate your PhenoAge, you should get what's called a chronological age, which is like what your calendar age is, and then what's called a PhenoAge, which is kind of your biological age. If your PhenoAge is lower than your chronological age, you're in good shape. If it's higher, then that means you have a higher likelihood of developing disease, or maybe you already have diseases, and lower likelihood of being disease-free. Possible increase of all sorts of diseases, and even chance of death, depending on how bad it is, so to speak. Here are the 9 markers. I'm not going to go into them in detail. Again, the calculator's on our website. You can plug in these 9 markers. Just to use an example of how you can plug it in. So this is kind of how the calculator on our website looks like. Here's the 9 markers I was telling you about, and what you need to do, you kind of go through them one at a time, and for example, the first one here is albumin. So you kind of look up albumin on your report. You plug the albumin score in. Creatinine, you kind of look that up on your report. You plug that in, and so on and so forth, all the way down. You put in your date of birth, the date of the blood test, and then your email address, and you should get a report right there and then, but also emailed to you. And this is just finishing up the other 8 markers. All right, so this is what the report will look like. So in this particular case, this person's chronological age was almost 63 years of age, having a date of birth in 1957, and the blood work was done, in 2020. But however, this person had really good biomarkers, and you could see that according to this algorithm, according to this one measurement of biological age, this person scored almost 16 years younger than their biological age, than the chronological age. So their biological age seems to be in 15 years or 16 years younger than their chronological age. It's not that simple. I mean, there are a lot of caveats to that. There's no perfect BioAge test, but this is one tool that can be used. All right, moving on to subjective. During the clinical trial, we used a system called the CASTOR, where we presented surveys to patients, and there's 2 main surveys that we obtained: the short form 36 and the WOMAC, the Western Ontario McMaster University Arthritis Index. So typically, the WOMAC is used for arthritis, knee and hip arthritis. We thought that rapamycin had a decent chance of improving those scores. We don't know yet because we haven't finished the trial, but maybe we can find some promising trends. Likewise, the SF36 has 4 questions over 8 domains, 4 times 8 is 36, looking at physical functioning, pain, general health, social functioning, mental health, things like that. And it kind of gives you an overall assessment of quality of life. Again, the idea was, or the hope was, that we would see an improvement in quality of life in the rapamycin group versus the placebo. All right, so part 2 here, I'm going to jump into DEXA scans and some of the other tests that we're looking at. These measure more, even though I kind of touched on efficacy when I was talking about the PhenoAge. But we're going to be digging a little bit more deeper into the other tests that are available or that we're looking at. So a DEXA dual absorption X-ray is looking at the classic use or the conventional use of a DEXA is to look at bone density. The DEXA scan that we use is a little bit different, and I'll explain that a little bit when I show a report. But basically, you get several measurements with the DEXA scan. You get lean mass, which is basically non-fat. You get the visceral adipose tissue, and the less of this you have, the better. And this is the relatively small amount of fat that's hidden around the organs, and then you get a whole bone mineral density content. Again, it's not 100% accurate. Like, you can't diagnose osteoporosis, for example, with it, but it does correlate very well with the diagnostic bone mineral density. And there's reasons why. Why don't we just throw the bone? There's reasons why the BMD itself is a very specific test. It looks at very specific parts of the bone. I think it's L4, the lumbar, for the 4th lumbar, I think, or maybe the 5th, and then I think the left hip. And so, but that requires a technician to kind of isolate it and things like that. What we do instead, for the wellness test, this DEXA wellness test, the body composition, is just look at the entire bone density. And of course, it's very rare for somebody to have a really good bone density on their entire bone density, but have a really poor DEXA scan. I mean, it just doesn't work that way. There is a certain, I think it might be plus or minus 10%, even though sometimes I've seen it spot on, but it could be up to 10% off compared to the diagnostic bone mineral density. So this is what, if you get your DEXA scan from one of our partners, DEXA Fit, then this is how how the reports will look. You'll have a measurement over time. You'll have the fat mass in pounds in black, and you'll have the fat free in pounds in this magenta, this pinkish color. And the hope is that over time your lean mass or your fat free mass goes up, and because you're building muscle, working out, and your fat mass goes down, assuming you're not already at a healthy weight. There is a hint of visceral fat on here as well, and this is something we're tracking with our participants. It's one of our primary outcomes. This is also part of the report. This is for the bone density, and here you'll get I believe a T and a Z score, but it's for total. So this particular participant has a very good T and Z score. 0 is considered to be normal, like average. 1 is considered to be 1 standard deviation. This person is almost 2.5 standard deviations higher than their age group, and 1.5 standard deviations higher than a healthy 30-year-old at optimal bone density. We don't expect to see any changes in the bone density. You never know. Maybe rapamycin and autophagy is benefiting, or maybe even not benefiting bone. We won't know until we run the numbers. But that's part of the DEXA scan. All right, GlycanAge. The GlycanAge is a little difficult to explain, but the basis of it is that we have these immunoglobulins. There's several types. There's G type, M type, E type, A type. This particular one looks at the G type, the IgG, immunoglobulin G. And on the immunoglobulins, there are these naturally occurring glycans. The patterns of glycans have been thought to be important as markers of health. So it's thought that, honestly, we're not sure what causes what. We're not sure whether glycan patterns affect aging or affect diseases, or do diseases and aging affect glycan patterns. That's still being worked out, but there is an association between these glycan patterns and stress, aging, menopause, and then of course, in the case of rapamycin, which we're hoping that would improve the immune system, we're hoping to see an improvement in these glycan patterns. We'll measure it, we'll run the numbers, and we'll see what we can get. But that's the basis of GlycanAge, is looking at these molecules, these glycan molecules attached to the IgG proteins in the body. And you'll get a result like this, where again, it's very similar to PhenoAge, where you have a chronological age and you have a biological age. In this example, this person was, their GlycanAge showed them to be 5 years older. Again, it's measuring something different, let's say, than PhenoAge, something different than the methylation. So it's yet another marker that we can look at to help determine somebody's health status. You'll also get, anti-inflammatory indexes. Again, it kind of breaks it down by different glycans and different patterns that they're looking at. And again, it kind of gives you your score compared to others. Same thing with, there's an inflammation index here as well. So hopefully, when our participants get their reports back, they can read through it carefully, and there are some explanations of what these mean and what people can potentially do about it. All right, I want to jump into the one of the more exciting and potentially promising BioAge test called based on methylation. A lot of people call this epigenetics or epigenetic clocks. And the reason it's called epigenetics, because if you remember from any kind of biology class, you have your DNA sequence is made of ACTs and Gs. Those are kind of the nucleotides, and you've got a double helix. This is not supposed to change. So you're born with 23 chromosomes, and you'll die with 23 chromosomes. For most people. For almost everybody, 99.9% of people on Earth. There are some exceptions, of course, but I'm not going to get into those exceptions right now. But my point is, you're born with a certain amount of DNA, or certain patterns of DNA, and you'll basically die with those as well. Most people will, unless we have some kind of genetic engineering or something. But that's a whole other can of worm I don't want to open right now. So the question is how do you take the static information and make it so that your cells can function in the real world? And it's part of that control is thought to be these methylation patterns. So by methylation, there's the methyl loop CH3, that are attached to various different nucleotides on the DNA, and the body attaches them and kind of detaches them. There's various different mechanisms on how that happens. It's very complicated biochemistry. So I'm not going to go into that, other than to say, your body kind of uses this methylation to either turn genes on and off, or up or down. It's not a light switch. It's not on or off. It's more like a volume dial, up or down. And it's thought that when you're young and healthy, these methylation patterns are very robust, doing exactly what they're supposed to be doing, so that your liver functions like a liver and your lung functions like a lung. But as you get older, these patterns start to degrade, and your liver, doesn't necessarily function as a healthy liver should, and your lungs don't necessarily start functioning as a healthy lung should. And we think part of that is because this methylation pattern starts to break down. But there are tools that we use. This particular partner that we use, called TruDiagnostics. Last time I checked, they're measuring about 900,000 of these methylation markers, and they use subsets of these markers to calculate various different algorithms. And these algorithms are trained on all sorts of data sets and things like that, to provide various different insights on somebody's epigenetic or methylation age. So you should when you review a report, it's going to be pretty comprehensive from TruDiagnostic. They do a pretty good job of explaining it, and hopefully enough to answer questions. All right, so other tests that we look at, microbiome. The Thorne is the partner that we've identified, and very grateful for their help. Patients were participants were asked to do a baseline, and then a 12-month. And the idea is rapamycin might have some benefit on the microbiome, and that's potentially part of what gives us benefit. The rapamycin, its benefit, in addition to mTOR and autophagy, and microbiome might be at play here, so we're measuring it. GeroSense is an algorithm used by our partner Gero. It's a algorithm that uses measurement data mostly from like Apple Watches. So I think it's only available to Apple iPhone users. Again, we're looking for that data from participants to see if there's trends. And then Jonathan Ann, he's an academic dentist. He's very interested in looking at rapamycin's benefit on oral health. There was some really interesting mouse data, and there's some really interesting anecdotal evidence or anecdotal data from a lot of rapamycin users who are kind of using it on their own, that it can help potentially rebuild bone, reduce periodontal disease, and that's a disease that's very hard to reverse in classic dentistry and conventional dentistry. So if that pans out, if this can give us hints that some people are having benefit, then that could pave the way for very promising dental treatments based on mTOR and rapamycin. All right, so the Thorne, I mentioned the Thorne. You'll get a participant will get a report, and kind of breaks down gut health. It looks at various different markers. This particular participant, was told that they have a low digestion score, and that's based on a variety of different factors that was looked at. They have scores for digestion, and it kind of talks a little bit about what the rationale is in this case, an overabundance of a particular type of bacteria, and it kind of explains why. And this particular example, the inflammation score is also high. It's kind of based on also either bacterial patterns or various different proteins or liposaccharide patterns that are found in the stool. All right, so biobanking. So I'm gonna move on to biobanking. So biobanking is the idea of taking somebody's blood sample and putting it in storage to save for later. And the idea is, there are new diagnostics and new tests that are coming to the market all the time, and we're hoping to store the blood and work with partner labs to try to identify up and coming biomarkers. And the idea is, we have this blood in storage, and we can pull it out for our partners and have them run analyses. So it's a chance to kind of extend the usefulness of our clinical trial by storing the blood and making the blood accessible to pretty much any lab that wants it, Of course, there's a screening procedure and things like that. We're not just going to hand out blood samples. There's a limited supply. But we're very excited about the possibility of being able to find some unique insights from the biobank. All right, moving on to part 3, the post-trial. This involves the unblinding process and also where participants will end up after their participation in Pearl. So for the unblinding process, we're going to wait until the last person finishes their 12 months. And we've already had several participants finish. I think by now, we're in September of 2023. By now, about half participants have finished. We're waiting for about the other half to finish off by the end of 2023. So we can't unblind until the last person's done and we've even finished analysis, again, because we don't want to bias the analysis of the data by unblinding too soon. So once we finish the trial, we'll be notifying our participants on which group they were in, whether they're on the placebo, the 5-a-week, or the 10-a-week. And we made a promise to our placebo group participants that if you are in the placebo, sorry you didn't get the rapamycin, so we'll make it up to you with 48 weeks of rapamycin treatment through AgelessRx, and we'll cover the cost for that treatment. Again, this is something, if you were in the placebo group, this is something that will be, the research team will reach out to you and help you coordinate what the procedures are. All right, thank you very much. If you are part of the Pearl trial or if you have questions about it, the best way to get hold of us is you could look at our updates on AgelessRx.com/Pearl. We'll be posting these videos and other webinars and things like that we've done there. Of course, you can reach out to the research team, research@AgelessRx.com. If you'd like to speak to somebody live, there's our phone number for general AgelessRx. The people who answer the phones may or may not know everything there is to know about the Pearl trial, so they'll probably refer you to one of our research staff. So honestly, to contact us, the best way to get hold of us is research@AgelessRx.com. Thank you very much. And for those of you who are in the clinical trial, it can't be thankful enough. Without your participation and your help, we wouldn't be able to move forward on this groundbreaking study. Thank you.